Welcome back friends, welcome back in another video tutorial for microbiology with Chagnik. So we have been discussed about various method of protein sequencing, right? In the very first video we have discussed how we can determine the amino acid composition. Then we have discussed the N-terminal detection with the help of Sanger's method as well as Danside chloride. Then we have discussed about the advanced degradation, that is the protein sequencing method. Then we have learned that a protein of less than 50 amino acid can be sequenced well, right? So we have learned how we can convert a large protein into small polypeptides, right? With the help of what? With the help of various type of peptides, right? Then in the previous video we have talked about how we can break the disulfide bond as well as the non-covalent bond. Now it's time to understand how we can determine the amount of protein in an unknown sample, and this process is known as protein assay. Okay, so suppose you have a solution and you want to measure the amount of protein in that solution. So if you ask me, I will suggest you the simplest type of method. I will suggest you to go and use the spectroscopy to measure the absorbance at 280 nanometer. Okay, then you will ask me that why at 280 nanometer. So I will tell you that in your protein, if there is any kind of protein in that particular solution, then in that protein there may be some aromatic amino acids, right? And aromatic amino acids are phenylalanine, tyrosine, tryptophan. So this aromatic amino acid have absorbance at UV range, especially at 280 nanometer. That is why protein absorb UV light especially at 280 nanometer and give you a absorbance value. Okay, so especially due to the presence of aromatic amino acid as well as for total concentration, protein will absorb the UV light. Now there are other various type of biomolecules that may be present in your solution, and they may also absorb UV light at 280 nanometer. For example, nucleic acid. Nucleic acid absorb UV light at 260 nanometer. Okay, they may also give you absorbance at 280. That is why, although it will not be the maximum absorbance, but still it will give you some value. So that is why it becomes very difficult to understand whether the reading that we are getting is for the protein or not. And for that reason we have moved on from the normal spectroscopic technique to the colorimetric technique, where we are using various type of reagent that will going to form a complex wheel protein and create a colored solution, so that we can understand whether the protein is present or not, and then we will quantify it with the help of various colorimetric technique. So after that we will going to create a graph with y axis there will be the OD values, that is the absorption values, and in x axis we will take the amount of protein. There will be some standard values, that is for known concentration of purified reference protein, and a unknown value, and by simple extrapolation technique we will find out the concentration of protein. Suppose all these red point in this diagram are for protein of known concentration, so we have drawn this, and along with that we have also used another tube where there is a unknown solution. So unknown solution will also give you a point in the graph. Now by simple extrapolation you can find out what is the concentration of protein of that unknown solution. That is the idea. So first we will going to talk about what are the reagents that we usually use in the colorimetric measurement of protein. So first I hope you understand why we have moved from the normal spectroscopic technique to the colorimetry, to understand whether protein is present or not, right? So now let's move on this. So previously we are doing simple this type of techniques where we measure absorbance at 280 nanometer with protein containing unknown solution, and it will give you absorbance due to presence of aromatic side chain which have strong UV light absorption, right? But now I have told you that as nucleic acid also have absorbance in that range, so they may also give you some reading in that particular absorbance. So to confirm yes protein is present, we will going to add reagents, and depending upon the reagent we use, there are three standard methods for protein estimation with the help of colorimetric technique. One is Biuret method, which is less sensitive, then Folin method, which we also call as Lowry method, and at last Bradford method. Okay, so in Biuret method, there is a formation of complex between the cupric ion and the peptide bond that will gives us absorbance, although it is not highly sensitive because it has no high extinction coefficient. What is extinction coefficient we have talked about in our spectroscopy lecture series, you may have also watched. So that is the idea of Biuret method. So first, it is not that much sensitive. Why? Because it do not have any kind of high extinction coefficient, and what is happening here, there is a complex formation occurs between the cupric ion and the peptide bond of protein. Now let's talk about what happens in case of Folin method. Folin method, also known as Lowry method. Here first, cupric ion peptide complex forms, that also observed in case of Biuret method, but the twist is, which makes Folin method a very useful method, that is, along with that, a complex is formed that is known as phosphomolybdate complex, and this phosphomolybdate complex is especially due to presence of aromatic amino acids. Okay, and that makes this Folin method more sensitive than Biuret method. And at last, there is a Bradford method where we are using chromacy brilliant blue, and this chromacy brilliant blue will going to react with the free NH2 group of the amino acid chain. Okay, so that is the Bradford method. So in Biuret method, we are getting complex of cupric ion and peptide bond. In Folin method, cupric ion and peptide bond along with phosphomolybdate complex. And in Bradford method, chromacy brilliant blue we are using that interact with the free amino group in the side chain of the amino acid. Okay, so that is the idea. So let's move on to the very important part, that is, I have told you that 280 nanometer absorbance of protein is due to aromatic amino acid, right? So now along with that, there are two more wavelength where also protein gives a good absorption. One is at 260 nanometer, and this absorption is due to the presence of disulfide bond between two cysteine. And another type of absorption that is observed is at 205 nanometer, and this is especially due to the presence of peptide bonds. Okay, but there are some kind of problems that we used to face when we will use colorimetric technique or any kind of spectroscopic technique for concentration of protein measurement. What these kind of problems are? First of all, the conditions that determine the tertiary structure of protein, for example pH value, for example the buffer, for example the reducing agent, will going to affect the absorption of protein. Because try to understand, if pH buffer or reducing agent, the amount of pH buffer and reducing agent is somehow changed, then the charge of the protein as well as the tertiary structure of the protein can be changed, and it may affect the absorption. Okay, more or less we can get a OD value. Okay, OD value, which is the absorption value. Now the idea is how we will going to determine the concentration of the protein from that OD value, because our main goal is to what? Our main goal is to understand or is to find out the concentration of the protein present in that unknown solution, right? So there is a formula that tells us that one OD value at 280 nanometer is equal to 1 milligram per milliliter of protein. So now suppose you have get a OD value 7, okay, at 280 nanometer. That means what? That means you have protein of 7 milligram per milliliter concentration. Now let's try to think about this. You have taken 4 milliliter solution in that particular test tube. So in 1 milliliter you have 7 milligram protein, then in 4 milliliter what you will have? You have 28 milligram protein. So that is the idea how you will solve. But it is not that much easy, because as I have told you, that nucleic acid also absorb UV light at that particular nanometer, right? Although it is not that much maximum as it used to be in case of protein, but still it absorb and it will hamper the OD value. So to determine the most accurate protein concentration, you have to use this formula, that is 1.55 into the absorption at 280 nanometer and minus 0.76 into the absorption at 260 nanometer. The value you will get, suppose again you have get a value of 6 OD here. So according to this formula, then you have 6 milligram protein per milliliter. Okay, so that is the idea. That means if I am going to sum up this, you have a test tube where you have unknown solution containing protein. What you have used? You have used, let's say, Bradford reagent. Okay, so entire solution becomes blue. Okay, so now you have used it into colorimetric instrument and you have gotten a value by calculating at the absorbance at 280 nanometer and also at 260 nanometer. What you will do? You will multiply 1.55 with the value of absorption at 280 nanometer and you will also multiply the value of absorption that you have gotten in 260 nanometer with 0.76. Okay, then you will subtract these two values. The value you will get is still is in absorption, right, or it is still a absorption value. Now what you will do, you know that one absorption unit is equal to 1 milligram per milliliter of protein at 280 nanometer. The value you will get, suppose you have gotten 8 absorption unit, so the concentration of protein in your test tube is 8 milligram per milliliter. Okay, and now let's say you have used 3 milliliter solution in the test tube, so then 3 into 8, 24 milligram is your amount of protein in that particular test tube, right? So that is how you can measure the protein. So I hope this video will be helpful for you. Thank you.